Hello, future doctors. If you're ready to absolutely master the cell chapter for your NEET exam, then you are in exactly the right place. In this one complete explainer, we're going to cover everything from the ground up, making sure you have all the key concepts totally locked in. So here's our game plan. We're going to start with the basics, the cell theory. Then we'll jump into the big divisions, prokaryotes versus eukaryotes, and plant versus animal cells. After that, we'll take a really detailed tour of all the organelles, and then we'll wrap it all up with a high-yield revision section to make sure everything sticks for exam day.
All right, first up, life's fundamental unit. Before we can get into all the complex machinery inside, we really need to get a handle on the core idea that defines every single living thing.
This is such a great question to start with, right? I mean, think about the incredible diversity of life on Earth. Everything from a tiny bacterium to a gigantic blue whale. What in the world connects them all? Well, the answer is the cell.
And this right here, this is the definition you've got to lock in. All life is made of cells. It doesn't matter if it's a single-celled organism like an amoeba doing everything in one little package, or a super complex organism like us. It's all built from this one fundamental unit.
What's really cool about this timeline is seeing how scientific understanding comes together piece by piece. So, in 1838, Schleiden figures out that all plants are made of cells. Just a year later, Schwann sees the exact same thing in animals. But they were still missing a huge piece of the puzzle. Where do new cells even come from? It took until 1855 for Rudolf Virchow to add the final crucial piece with his famous statement, omnis cellula e cellula. All cells arise from pre-existing cells. This is a super important point for your exam.
Okay, Section 2, Prokaryotes versus Eukaryotes. Now that we know the cell is the basic unit of life, let's look at the first massive split in the living world. This is the core difference between simple cells, like bacteria, and the more complex cells that make up you, me, plants, and fungi.
Think of it like this. A eukaryotic cell is like a huge organized factory. It's got different departments or organelles, and each one is in its own room with walls. We call this compartmentalization. A prokaryotic cell, on the other hand, is more like a single, open workshop where all the tools and machines are just mixed together in one big space.
So, for your exam, you absolutely need to remember these unique prokaryotic features. Their genetic material just kind of floats in a region called the nucleoid. It's not wrapped up in a true nucleus. They have 70S ribosomes, which is different from eukaryotes, which have 80S. And they often have some pretty cool extras, like plasmids, these little rings of DNA, and even gas vacuoles to help them float.
All right, on to Section 3, plant versus animal cells. Now we're zooming into the eukaryotic world to look at the second great divide. We're going to compare the two classic cell types you have to know inside and out for NEET, plant cells and animal cells.
This table is pure gold for your revision. Seriously, let's walk through it. A plant cell has a tough, rigid cell wall made of cellulose for support, which animal cells just don't have. Plants have plastids, like chloroplasts for making food. They also usually have a huge central vacuole to maintain pressure. Now, on the flip side, an animal cell has centrioles, which are key for cell division, and they almost always have lysosomes for breaking down waste. Make sure you know these 5 differences like the back of your hand.
Okay, let's put on our imaginary hard hats, because we're about to take a detailed tour of that eukaryotic factory floor. We're going to go through each major organelle and look at exactly what it's made of and what its job is.
Every single eukaryotic cell, plant or animal, is wrapped in this incredibly important dynamic barrier, the plasma membrane. And it's not just some simple wall. Think of it as a very smart gatekeeper.
So how does this gatekeeper actually work? Well, the best way to picture it is the fluid mosaic model. It describes the membrane as this fluid, sort of oily sea of lipids, with big protein molecules floating around in it like icebergs. This fluid nature is so important. It lets the cell grow, change its shape, and even release substances.
And this is where its job as a gatekeeper really shines, controlling traffic. You've got passive transport, where molecules just flow from high concentration to low. It's like rolling downhill, so it doesn't cost any energy. But then you have active transport. This is like pushing a boulder uphill. The cell has to spend energy, in the form of ATP, to force molecules to go where they don't naturally want to go.
Now, some organelles don't just work alone. They're part of a team, like a coordinated assembly line inside the cell. We call this the endomembrane system, and it includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles.
First up is the ER, which is this big network of membranes. So what's the difference between rough and smooth? Well, the clue is right in the name. The rough ER is covered in little dots called ribosomes, which makes it look rough, and its job is to make proteins. The smooth ER, you guessed it, doesn't have ribosomes, and its job is to make lipids and to detoxify poisons.
A great way to remember the Golgi is to think of it as the cell's post office. Proteins and lipids arrive from the ER like mail. The Golgi then modifies them, sorts them, and packages them into little bubbles called vesicles, so they can be shipped off to their final destinations, either inside or outside the cell.
Meet the cell's recycling crew, the lysosomes. These are little sacs filled with powerful digestive enzymes. Their job is to break down junk, like old, worn-out organelles, food particles, or even invading bacteria. They're absolutely essential for keeping the cell clean and healthy.
And now for the one you've all been waiting for, the famous powerhouse of the cell, the mitochondria. This is where the magic happens, where the food you eat gets converted into the cell's main energy currency, ATP.
For your NEET exam, these features are critical. It has two membranes, and the inner one is all folded up into these things called crista, which creates a huge surface area for making ATP. But here's the most important part. Mitochondria are semi-autonomous. They have their own DNA and their own 70S ribosomes, which means they can actually replicate all by themselves.
Now we arrive at the very heart of the eukaryotic cell, the command center itself, the nucleus. This is where the cell's genetic blueprint, its DNA, is housed and protected.
So the nucleus isn't just a blob. It's protected by a double membrane called the nuclear envelope, which has tiny pores to control what gets in and out. Inside, there's a dense spot called the nucleolus. That's where ribosomes are born. And all the DNA is neatly packaged with proteins into a material called chromatin, which coils up super tightly to form the chromosomes you can see when a cell divides.
Okay, we have been deep inside that cellular factory. Let's zoom back out for a minute and pull everything we've learned together into a powerful, high-yield summary that's perfect for your last-minute revision.
This right here is your ultimate cheat sheet for the exam. Let's run through it one more time. Ribosome, protein synthesis. Mitochondria, the powerhouse for ATP. Golgi, the post office for packaging. Lysosome, the suicidal bag for digestion. And the smooth ER, lipid synthesis and detox. You have got to know this table inside and out. And to wrap things up, here's a final challenge. A classic NEET-style question. Which of these is not part of the endomembrane system? The trick is to remember that the system is all about teamwork. The organelles have to work together in a sequence. The ER, Golgi, and vacuoles are all part of that team. The odd one out is the peroxisome, which kind of does its own thing. Understanding these relationships is what will set you apart. Good luck with your studies.